Hello, everyone. Welcome to the October edition of the CNS Journal Club podcast. My name is Jeffrey Traylor, and I'm a resident at UT Southwestern Medical Center in Dallas, and I'll be the moderator for today's episode. Today, we'll be talking about Posaconazole for glioblastoma, and we're joined today by our author from Penn State, Dr. Mansouri. Dr. Mansouri, would you mind introducing yourself for us?
Sure thing. Hello, everyone. I'm Alireza Mansouri, Assistant Professor of Neurosurgery at Penn State.
And we're also joined today by Dr. Ganesh Rao. Would you mind introducing yourself for us?
Sure. I'm Ganesh Rao. I'm the Chair of Neurosurgery here at Baylor College of Medicine in Houston, Texas.
And finally, Dr. Vega, the co-chair of the CNS Journal Club podcast, is also joining us today.
Thank you for the introduction, Jeffrey. And again, my name is Raphael, and currently the director of the Brain Tumor Center here at Beth Israel Deaconess Medical Center in Boston.
Wonderful. So, Dr. Mansouri, just to start us off, would you mind just giving us a brief overview of the article, the protocol in this case for your paper in neurosurgery?
Sure thing, Jeff. So this is a phase 0 clinical trial, mainly utilizing microdialysis catheters to interrogate the blood and tumor interstitium. So this is based on exciting preclinical data that we had in vitro and in vivo that we found posaconazole was able to inhibit GBM tumor cell growth in mice. And before we launch into large-scale studies looking at overall response rate and survival, it's critical to establish whether these drugs even cross the blood-brain barrier, accumulate in the tumor at a sufficient enough concentration, and do what we expect them to do from a biological perspective. So that's why taking the strategy we thought would be useful. So patients who are deemed to have a GBM, who are going on to surgery and are stable enough to take a drug for several days before surgery up until we have steady state dosing, would be enrolled in the study, and they would take the drug. And at time of surgery, the tumor is taken out, and we try to sample both contrast enhancing and non-enhancing regions, send it to the lab with mass spec, try to look at drug concentration and also lactate pyruvate concentrations. And at the completion of the surgery, we insert microdialysis catheters. If there's any enhancing portion left, we put it in that, and in addition to non-enhancing portions. If there's no enhancing segments left, we just put the catheter in the non-enhancing portion. And what this allows us to do is over the next 24 hours, we can measure the concentration versus time profile of the drug and lactate and pyruvate, so kind of giving us a neuro-pharmacokinetic profile as well.
Wonderful. Thank you for that summary. Let's go ahead and get started with a discussion with our guest faculty. Dr. Rao, did you have any questions for Dr. Mansouri?
Yeah, it's a very interesting concept. And I wonder, some of the biology behind it's very interesting too. I wonder if, Dr. Mansouri, you can maybe describe in a little more detail how cancer cells exploit the Warburg effect to survive in hypoxic conditions, and then how this sort of aberrant glycolytic metabolism can be targeted therapeutically.
Sure thing. As we all know, any malignant cancer cell tries to utilize the Warburg effect to survive under hypoxic conditions. So cancer cells can generate more than 50% of their ATP from glycolysis rather than oxidative phosphorylation. And they do this by enhancing glucose flux, and therefore this allows them to produce ATP faster per glucose molecule, particularly on these hypoxic conditions. For GBM, it seems to leverage hexokinase 2 as a key component of the Warburg effect, and it has an important role in glucose flux into glycolysis or the pentose phosphate pathway, and its transcription is tightly regulated by HIF1 alpha, glucose, insulin, glucagon, among other molecules. And previously, our team had shown that downregulation of HK2 inhibits aerobic glycolysis while promoting oxidative phosphorylation, as shown through increased oxygen consumption, decreased extracellular lactate, and increased expression of genes involved in oxidative phosphorylation. So given this strict dependence of GBM cells on HK2 as a pivotal enzyme and the gene networks associated with it, we found it to be an ideal therapeutic target.
Very interesting. Why, and for the audience that may not be so familiar with brain tumor treatment, can you describe why the blood-brain tumor barrier remains a challenge for primary brain tumor treatment? I think a lot of people think they just because you can get contrast enhancement, that suggests enough of a disruption to permit therapeutic agents from entering the microenvironment, but that may not always be the case, correct?
Absolutely. We have done a great job curing brain tumors in dish and in mice for many years now, but unfortunately, not many of these have translated in the clinical setting. And there's many issues that are at play, but a big part of this is the blood-brain barrier. If you think about it, our best drug that we have against glioblastoma, temozolomide, at the peak brain to plasma ratio, it is only 20%. And as we know, contrast enhancement in essence basically means leaky vessels. It doesn't necessarily mean that it's a disrupted blood-brain barrier. And when we use gadolinium as our contrast agent, the particular region that is permeable to a compound, to gadolinium, it means that it's permeable at best to a compound that's as big as gadolinium. So things that are bigger may not necessarily get across. And this doesn't take into consideration the many efflux pumps and other mechanisms that cells have for spitting the drug back out. So therefore, short answer to your question is no, it doesn't necessarily mean that just because we have GAD enhancement, we have reliable blood-brain barrier disruption. However, it is a starting point. And as we know, at least some portion of the blood-brain barrier may be leaky in this area, and it can be a starting point for our studies.
Can you give us a sense of the success of preclinical studies with PCZ? And the reason I ask is that it's sort of a leap. I think people, most investigators, or most practicing physicians, I should say, don't always appreciate the amount of work that has to go in the preclinical setting before you take trials to humans. So what kind of threshold do you have to cross, and what was the specific success you had here before you were able to go to a phase 0 trial?
Yeah, so we took a very stepwise approach. First, it was an in silico screening strategy with silencing RNAs, and then in vitro testing against not just U87 mouse models, tumor cell lines, but also glioma stem cell lines, 2 of them. And then finally moving on to in vivo, again, using multiple cell lines to really definitively prove our case. So after we found our drug candidates using this in silico approach and in vitro methods, we moved on to mice. And after that, we were able to show that mice bearing glioblastoma and treated with both ketoconazole and posaconazole at a dose that would be clinically translatable and safe in humans, increased their survival, reduced tumor cell proliferation, and decreased tumor metabolism. And in addition, we were able to histologically show that treatment with posaconazole resulted in an increased proportion of apoptotic cells. So again, we've cured mice a lot from tumors. This doesn't necessarily mean it translates, but we felt confident enough that we have all these data lining up that the next logical step to see if we can emulate this in humans.
Great. I guess my next question is really related to this drug and the way you got to it. So I'm curious to know how, you took a drug that's been out there in the pharmacological world for a while and have sort of repurposed it. So I guess, can you give us a little more background on how you came to find this drug, and do you think there's an opportunity for repurposing other drugs for the treatment of GBM?
So this was a build-up on a prior work done in the lab that showed the importance of hexokinase 2. And it's been also shown that it's not strictly just what role it plays in glycolysis, but also the network of genes that it interacts with. So the steps that we took was basically try to treat different cell lines with siRNA pools against HK2, extract their RNA, and assess the genes that were differentially expressed, either upregulated or downregulated in siRNA-treated cell lines versus control. And then from that, we identified pathways and then performed a connectivity map analysis to cross-reference this against the database of FDA-approved drugs that would affect these particular pathways. Among that, we found 15 candidate drugs. 10 of them were available for testing. And then these drugs were then tested. We performed a viability assay, looking at the viability of tumor cells, but also normal human astrocytes and neural stem cells. And we want to make sure that we pick a drug that does kill tumor cells at a reasonable concentration, but not human astrocytes and stem cells. From that list, ketoconazole actually emerged, and we knew from the literature that mebendazole is another azole that has been tried as a repurposed drug. So we picked Posaconazole because it is a more friendly drug, less drug-drug interactions, some pretty good data on blood-brain barrier penetration. So then we tested that, and we showed that the EC50, the effective concentration at which half the tumor cells can be killed, was again within the safe, clinically translatable dose against tumor cells, but the EC50 would be much higher to kill normal human astrocytes and neurostem cells. So we deemed that this would be a more translatable drug. So that's how we got to it. And then in terms of opportunities, a major challenge with glioblastoma is the many aberrant metabolic and genetic pathways that have been associated with it. But this also means that we have the opportunity to exploit these. So hexokinase 2 is just one of these central pathways, but there's many out there. And there's plenty of examples on clinicaltrials.gov. If you do a search, you'll find plenty. So metformin is one that has many trials registered for it. Mebendazole, as I mentioned, disulfiram, SSRIs. Some of these are at the clinical stage, so phase 1-2 trials, and some of them are preclinical. But it's very exciting. And, as surgeons, we have the opportunity to really be at the forefront of that by interrogating the brain and its environment with these microdialysis catheters and really trying to see if they do what they're supposed to do.
I did have one follow-up question. Since I know that this is an anti-metabolite, and with the most recent WHO update of 2021, that GBM is now defined by IDH wild-type status. Do you expect with this drug that there would be any different effect in IDH mutant gliomas, like a grade 4 astrocytoma, for example, based off of your data so far?
I think that's a great question. It's very timely, of course. I would certainly not rule it out, and this is something that we would need to look at. So we would be looking at the data of our IDH data of our tumors and see if there's any differential response. It's kind of hard because this being a phase 0 study, we only have 5 controls and 5 treated tumors. But as hopefully we get to a phase that we can expand it and look at this data. I would note, though, that HK2 plays a role at the very early part of cellular utilization of glucose before the beautiful Krebs cycle. But then again, it's also important to emphasize that posaconazole affects HK2 transcription and the many genetic and metabolic pathways associated with it. So it's not just direct inhibition of the HK2 enzyme.
I see. And then one last question from me. I saw in your protocol that you expected to see an increase to posaconazole concentration in contrast enhancing regions rather than non-enhancing. Is that just because that's the more metabolically active part of the tumor, or is there another reason?
That's actually a great point. So yeah, in our in vivo work, we had shown with mass spec that posaconazole does cross the blood-brain barrier, but it was difficult to ascertain really in which part, given that how small the samples are. Just by the notion that we have regional variability and the permeability of the blood-brain barrier in contrast enhancing and non-enhancing regions. This was our hypothesis, but I think that's also a very good point. If you envision that the non-enhancing parts are potentially lower grade components of the tumor, that's also a very valid point.
Wow. It's a very interesting, discussion. One thing I wanted to ask, and again, it's nothing too challenging, but just thinking about the microdialysis catheter placement, what would you think about doing this, like in the sense of like an open resection, and having the ability to deliver locally, and how that would be different? I don't know.
You mean intraparenchymal delivery of the drug?
Yeah.
So there are some groups looking at that. So I think out at Mayo Clinic with low-grade gliomas, it's really cool concept. The pore size of the catheter is bigger than ours. So ours is 20 kilodaltons, theirs is 100. And what they do is basically they expose the tumor, they put these catheters in different regions, and the candidate drug goes in. And as they resect the tumor, they take that part around the catheter out and look at the metabolome around that. And I think that's very interesting. But these things, if you've seen them, they're very tiny, very flimsy. So I can imagine it's very hard to operate around these things.
Of course, yeah. But it is an interesting concept, that's all. And I guess one of the last things, and this was for your own personal opinion, is what would you find is like the biggest barrier, in trying to really convert this into, in a clinical setting, like there's a lot of folks interested in these, dialysis catheters and whatnot, but it's we still haven't used them right in our clinical practice yet. So I'm just wondering if is this the one?
Yeah, there's many nuances. So what we didn't talk about is a lot of in vitro work that we did beforehand with the catheters. So for example, because these technically are plastic tubes, you have to make sure that the drugs are not going to stick to the plastic tubing, and they can't be too hydrophobic because they need to dissolve in solution. The pore size is obviously a factor, and the rate at which the catheter, the pump runs. So you can have anywhere from 0.1 to 5 microliters per minute. So, and then the temperature at which you store them. So all those things that have to be considered. So we had a whole set of experiments in terms of different rates, different storage conditions. And then finally we picked 1 microliter per minute. That's how we came to it. So not all drugs are amenable to that. That's number one. And as I mentioned, these are very fragile, so they can break. And we're learning more and more as we work with them how to troubleshoot them. So I think that's been a major barrier. And so for these patients, for example, it works out because the patients, after receiving 5-ALA, we do 5-ALA guided resection, so they need to be in the hospital for a good 24, 36 hours after the resection anyway. So this works out in terms of schedule. But if you otherwise would have sent the patient home, again, this doesn't work if you want to collect sample over 24 hours. Yeah.
No, I'm very excited about the future on this. So
yeah.
Yeah.
Hopefully we're successful.
Exactly.
Wonderful. Well, I think that's all the time that we have right now. If there aren't any more questions, I want to thank our author for joining us today, Dr. Mansouri from Penn State, as well as our guest faculty, Dr. Rao. Thank you so much for joining us today. I want to thank the listeners for joining in as well. We're all very excited about these results and really look forward to the results of this trial. And so I do want to add that this podcast activity is worth 1.5 CME and is complimentary to all CNS members and can be accessed on the online catalog. We hope that you'll join us next month for the November Journal Club podcast. Thank you so much.